So it's a great pleasure and wonderful for us that I get to introduce Arvind Murugan today. So Arvind started off on the string side of high energy theory. So he got his PhD with Igor Klebanov at Princeton, and the title of his thesis was Gauge Gravity Duality with Renormalization Group Flow and Reduced Supersymmetry. So a real physicist. No. This is purely by way of introduction. So after he did this PhD, he did a postdoc with Stan Leibler at the IAS and Rockefeller. And there he entered biology really with a beautiful paper looking at a new regime of kinetic proofreading. So this is a way to get some of the accuracy you could get with Hopfield's scheme, but to do it really much faster and facing some practical trade-offs. So then he did a postdoc with Michael Brenner looking at assembly of particles that can assemble into many forms. And in 2015, he started his group at UChicago. And since he's started really a program that I think is doing a lot of the most interesting and creative things in theoretical biophysics today. And in particular, he's really driven interest in what it means for a physical system to learn how a physical system can implement algorithms naturally, how it can store, retrieve, and really process information. It's beautiful work. Some of it he's going to tell us about today. And I'll just mention he's won a lot of awards, but most recently the Schmidt Polymath Award in 2025. So with that, I'll stop talking and leave it to you.
Thanks, Ben. And can you hear me out there through some microphone? Thank you all for coming out. It's a pleasure to be here at Yale. Like Ben said, actually my group, we work, I would say half the group works on biology in a way that a biologist would take seriously. But today I'm going to talk about the other half of what we do, which is certainly, for me, it's personally motivated by biology. I think it's relevant to biology, but is firmly on the more speculative end of things. Okay. And since it's a physics department, I presume you will stop me and ask questions rather than wait politely like some biology audiences do. So please don't be polite. Please ask questions right away. Okay, so what I'm going to talk to you about is a somewhat philosophical perspective on what it means for a physical or biological system, non-neural system to process information. So I want to begin with something very concrete to explain this perspective. Imagine I give you a challenge. Keep the temperature of this room fixed. One approach to solving a problem like that is what I would call an abstraction first or algorithm first approach, which is, say, go to a control theorist and ask them how to solve it, and they give you a abstract solution like this called a integral feedback loop, which says, measure the temperature of the room, subtract it from the base set point, integrate that error over time, and then actuate a heater or a air conditioner based on that. It's an abstraction first approach because this does not assume what physical substrate you're going to solve the problem in. You could do these operations with electronics, with molecules, with photonics, whatever, doesn't matter. It's guaranteed to work if you mimic this algorithm with some hardware. On the other hand, here is another solution to the same problem. Just a cube of ice in water at 0 Celsius. Okay? This seems almost trivial. It's just an ice bath. This will keep the temperature of this water fixed because if you add some heat, the ice cube will shrink. If you take out heat, the ice cube will expand. It solves the same problem. It's so familiar that we think it's a trivial thing, but actually it's a pretty non-trivial thing related to phase coexistence at first-order phase transitions. This Maxwell construction, was William Gibbs ever in this room? Maybe, maybe not. Okay, let's pretend he was. Okay, so it's a pretty non-trivial fact of things that those people did, that water has that property at that point, and it's a solution to the same thing. And you may think it's a bit silly. I'll give you some biological examples of this, but I just want to point out there are some downsides to this, many downsides to this. It's very particular. It's a particular solution. It only applies to first-order phase transitions. But there are upsides, which is what I want to essentially get across today, which is it's a much more robust and a simple solution. For example, On this side, you have to have a sensor, computational thing to integrate the error, actuator, all of that. In some sense, this side has all of that, except all rolled into one. You can't really separate which molecule is doing this and that and this wired up. It's just doing it all together. Another aspect is it's completely passive. It's not some power device that you need for it. And so, in fact, in the lab, if you ever wanted to keep things at 0, you'd obviously pick that solution. And my, some sense, viewpoint is we've sort of overlearned this kind of lesson when it comes to biology. When we ask, how does biology do something, we tend to look at it this way too much. And one example of this is literally that kind of control loop, where for a while in systems biology, people asked, can we get that? For example, in a synthetic biology context, how do you do that? How do you mimic every element of that? And the best work, in fact, took a while, 20 years of work. Mustafa Kamash managed to come up with a set of molecules that do each step in this. Okay? But in fact, his student, postdoc, Zechner, showed that in natural cells, there is the equivalent of the ice cube in water. I won't go into details, but essentially you can ask Ben about this. Many molecules do what's called phase separation, where they are in a dilute phase and a dense phase. And when molecules do that, the dilute phase concentration is clamped. If you add a few more molecules, the dense phase will grow, but the concentrations won't change. And in fact, in the cell, they showed, at least in some limited condition, that this solves a fluctuation problem for you. It helps a cell keep a concentration fixed. Okay? Another kind of problem people talk about is some kind of input-output problem. Okay? So imagine there's a cell that's trying to sense 4 aspects of its environment or 10 aspects of its environment, 10-dimensional inputs, and has to take one of 2 actions in response. Think of it as a neural network trying to classify cats from dogs, right? Except it's a chemical environmental signal. People say, well, okay, I know how to solve that problem because I know neural networks solve that problem. So they first write out a solution, it's neural networks. And then they say, oh, I know how to make neurons with molecules. I can get molecules, or maybe sometimes even cells, to act like linear threshold devices. And so I mimic each element of that, compile them all together, and then I've gotten a classifier of environmental signals. Okay? But in the end, what they've done with that is achieve sort of decision boundaries in this input space of images. Like, well, for a neural network, it would be images of cats and dogs. What you're really trying to do with the neural network is put decision boundaries that separate cats from dogs. Right? But here's a different way to do it. This is the phase diagram of water. The axes are temperature and pressure. And if you think of this as a 2-input, 1-output system, where the inputs are temperature and pressure, and the outputs are the phases of water, this is doing some kind of classification of that 2-dimensional input for you. For certain ranges of temperature and pressure, it's going to deploy ICE4 as a crystal. In other ranges, a different crystal structure. And that might seem, again, a bit silly, except in the cell, it's not water freezing. Instead, you have a multi-component molecular soup, many, many species of molecules. And as if you've gone to any biology talk in the last 15 years, you'll know they naturally form phase diagrams like this. So then you can solve a problem like this completely passively by just having the right phase diagram. And it'll sense all aspects of the environment that it's sensitive to. Does this make some sense? And in some sense, this is, I know, attention drops pretty fast in a 1-hour talk. And so if you get this point, you've gotten most of my talk, which is just the passive phase diagram of a multi-component system essentially can be a decision maker for you. You don't need circuits. Surely there are circuits, but you don't need it. Yes?
So in the first example, you explained how liquid-solid coexistence is helpful in stabilizing a parameter you might have otherwise used digital feedback for. Is here your point about these coexistence curves?
No.
Or you just want a 5-output?
Yeah, exactly.
Discrete outputs.
Exactly. So I'm using a phase diagram now in a different way, which is the coexistence lines are, I'm going to avoid them. I'm going to stay far away from them. Different environmental conditions, I want to deploy different functional responses. And that's another advantage of this framework, which is the output here, in this case it's crystals of ice, but with multi-component molecular systems, the output is going to be a condensate of some molecules. Often that is the functional output that biology is looking for. And so in some sense, the sensing, how do they say it? The sensing, the computation, and the response are all rolled into one, which is simply the process of water freezing. Okay. And so those are the advantages. And so in fact, this is a contrast that people have been drawing for 70 years now, say in computer science, which is general purpose computing has worked this way and has enormous advantages in allowing society to work. Like there are software engineers and then there are people who are making hardware and they don't need to talk to each other. The main downside is when you do this, you don't take advantage of the innate physics of what you're using. You don't let molecules be molecules, you force them to be logic gates in some circuit. And then you pay a huge overhead because of that. But if you're willing to give up on the general purpose aspect, the idea is there are advantages. And the claim is those advantages are attractive for me in the context of biology. I think evolution wants to just solve the problem it has with the hardware it has, not create necessarily general purpose solutions that will work on photonics or something else. The other community that's really interested in these days is actually for hardware for LLMs. All the ChatGPT queries we send, as you know, are expensive. And the idea is if we make the general purpose hardware a little more special purpose, you can save a lot of energy. In fact, when I'm not giving this talk to this kind of audience, we have a lot of workshops with people trying to do it for computing. But like I said, my own interest is that I think in extant biology, this is likely happening all over the place. It's just harder to spot these things. So 20 years of systems biology, we became good at spotting circuits because those are easier to spot to begin with. This kind of stuff is just the act of stuff condensing, right? Or the act of something gooping around on a membrane. It doesn't look like it's doing much, but it could be doing something. And of course, if you're trying to engineer something, maybe this compactness is useful. Today, though, I want to go beyond just that decision making to what we call sort of trainable decision making. The best way to communicate what we're after is this paper of Saeed Tawazwai back when he was at Princeton. And they showed the following. You take certain strains of E. coli and you raise the temperature on them. They act as if they are in an anaerobic environment. They express anaerobic enzymes, even though the oxygen levels have not dropped. And they argued that it's because in its natural environment, it sees a constant correlation, much like Pavlovian conditioning. Every time temperature goes up in your mouth, in mammalian mouths, 20 minutes later, those E. coli fall into your gut, and the oxygen goes down. So they saw this temporal correlation, like Pavlov, ring a bell, feed the dog, ring a bell, feed the dog. Then you ring a bell, and it starts salivating. And these E. coli, they argued, were like that. They put it in a lab in a fake anti-correlated environment and showed that you get a new correlation after that. Does that make some sense? Okay. The only thing is, this paper turned out it actually happened through evolution. Bacteria, and especially it's just breaking a correlation, it turned out it was through a mutation. But our question is, can you get this kind of behavior without any mutations? Okay. And just through sort of within a lifetime, or even if it's many lifetimes, like what theory does, just without any mutations. And the reasons are, you can ask people at Yale already about this, it's faster adaptation, and then there are also synthetic uses of this. Okay? And I'd argue today we don't even know what mechanisms for this could even look like. There are people who have proposed mechanisms that, again, take what neurons do and literally try to mimic it. And then you, again, end up with this huge overhead where you could engineer something, but it's hard to see it happening naturally. Okay? And so that's essentially what I'm going to tell you today. Not what actually happens in biology, but at least what would be the minimal ingredients you need for learned computation, if you wish. Okay? And I would argue this slide is still fairly general. After this slide, I'm going to give you my specific answers to these two questions. But anyone who tries to answer this broader thing needs to say, well, what is the substrate that's trainable? For the brain, we know there's a concept of, there's a reality of neurons and synapses they can make, and we know synapses can change, they're plastic elements. What is the equivalent of that at the single cell level, at the physiological level? And then what is the equivalent of, say, backpropagation on a computer or evolution on long time scales? that'll make the parameters of your system head in the right direction so you get Pavlovian conditioned, or you can have supervised training or something like that. Do these two questions make some sense? All right. So I'm essentially going to try and give you our answers to these that I think are minimal, but surely there are many answers to this. All right. So the first one, what can possibly be trained? The moment you think of supermolecules in the cell, well, you think about them interacting in some way. But the key thing is, unlike a neural network in the brain, the interactions in a network like this are not actually trainable. They are binding constants, which typically take many mutations to change something. And it's also not clear what kind of systems would actually be capable of many behaviors. And so the answer I want to give, essentially condensing some years of work, is that if you have networks that don't look like the usual systems biology networks, many molecular species that have binding interactions, but it's not sparse. Every molecule is typically interacting with a large number of other molecules. I want to claim that that is what we call a highly expressive substrate. I'll explain what expressive means in a moment, but that's essentially our answer for what's trainable. Okay. And the best way I can explain is with one simple example from Michael Elowitz's group, where in this BMP pathway, they found I would argue, a really simple example of a non-trivial computation. Okay? And here's the idea. There are two ligands in the system, green and blue, and there is some output. So and this heat map, the two axes are the two possible inputs. The output is some phosphorylation level of something downstream. And in this some particular cell type, you see a behavior that you could call an imbalance detector. The output is high if the two molecules are imbalanced in concentration. Now, that kind of non-trivial computation, it turns out, comes in from what molecules naturally do in this system. What happens is each of these molecules have a cognate receptor, meaning they really like to bind the thing that's right below them vertically. They have a strong binding constant to what's below them, but when they bind their cognate receptors, paradoxically, they do not signal much downstream after that. Okay? But they can also bind the non-cognate receptors, and if they manage to do that, then they signal strongly downstream. Which means if you have equal amounts of the two receptors, they will each sit on their own cognate ones, because they each like their own seats, and there'll be no activity downstream. But if there's a lot of blue and not much green, then the blue will eat up all of its cognate receptor and then be able to sit on a bit of the other receptor and then have strong signaling. Okay? So just this many-to-many binding across these receptors with sort of counterintuitive cross correlations in how they signal gives you this. Now, this is an anecdote, but it sort of asks what are the principles of such molecular interacting networks that lets you get not just this behavior, but a family of behaviors. Okay? And I want to show you, essentially, I'm going to just give you the punchline from a series of papers we've written since I was a postdoc of essentially what lets molecular systems do more, in some sense. And actually, I'm realizing since the physics audience, let me first quantify what I mean by do more before showing you. So in machine learning, people have come up with this concept of expressivity, okay, which is a measure of the range of behaviors a given system can do as you change its parameters. So for example, if you have a one-layer neural network or just a single neuron, by changing its weights, you can make linear decision boundaries in input space. And as you change its weights, you can make different decision boundaries, but they're always going to be linear. If you have a more complex, say, second layer one, or let's say you had polynomials and a quadratic one, then you can make more complex boundaries in input-output space. Does that make some sense? And as you get to deeper and deeper networks, you get more and more complex boundaries as you change parameters. So expressivity is a range of things you could approximate by changing all parameters. Okay? So our question essentially is, what makes physical systems expressive, put in this language? And one way to quantify it, don't look at everything in this picture for now, is we generate sort of training data from a sequence of teachers that are essentially neural networks or a family of polynomials of increasing degree. That is, we generate data like this, and then take each of these and challenge, or take one physical system, like I'm going to tell you, and challenge it to approximate these phase boundaries by changing all its parameters. Does that make sense? We take this molecular system, treat it as an input-output device, and ask, can it produce blue, like low output in these regions and a high output in the other region? And maybe this network can easily do this. Maybe it can do the next one, but maybe it's unable to do the one after that. So this axis here, the x-axis, is the complexity of the teacher, and the y-axis is the performance of the student. The student is a physical network. And if you take any particular physical system and you quantify it, it'll go up to some point and then start falling off. And the place where it falls off is how expressive is the system. Am I making any sense? Yeah?
What specific parameters are you training? Like, are they transition rates?
Great question. So that's exactly so. What I'm just going to show you is now assuming it's sort of an evolutionary perspective. So all parameters are up for grabs for a moment, meaning in this kind of molecular system, there are a bunch of binding constants and expression levels of those components. We train all of them and ask, how good are you? How far can you go? Okay? But then in a moment when we come to the learning part, I'm going to restrict some of those parameters, meaning the binding constants cannot change because you can't change that within a lifetime, and only change expression levels because you can change that in a lifetime, and ask, what's the range you have now? Yeah?
Maybe a related question, are you imposing any constraints on like how far from equilibrium the system can get or how much energy it can dissipate?
Yes and no. So today's talk, not really, but we have other networks where you take, meaning today's talk in some sense on purpose is equilibrium systems. Well, they're sometimes highly kinetic, but they're not non-equilibrium in the driven sense. But you can ask the same question for a non-equilibrium system at different levels of being driven.
One more question. So you're training, so you have these two-dimensional, like...
or in general, it's n-dimensional.
Yeah, and these are like the concentrations of the n species.
Yes, great question. So what are the axes? So for a neural network, the axes would be pixels of an image. For us, the axes, like in the BMP pathway, are going to be concentrations of different molecules, right? So typically, or it could be pH, it could be temperature, but they're going to be physical inputs. And so typically, they won't be very high dimensional. Its relevant ones might be 4 or 5 dimensional. Yeah.
Do you have this metric that sort of measures the point at which the fall off is? Do you care about what that fall off like approaches that sort of?
Oh, good question. I've not thought about that. We were just thinking of where it drops off as a measure of how expressive is it. Because this is saying essentially, so whatever that system is can approximate up to a 4th order neural network, but can't approximate anything beyond that. All right. So okay, that's a way to quantify it. What is the family of systems we considered? So it's essentially, in a way, a generalization of systems of this type, even though some of the work started quite a bit before. And to your point, it's actually in the spirit of like how much power is hidden in the simplest stuff. We are just considering just association. The simplest thing molecules can do is just stick to each other, fall apart. Okay? No activity of any kind, stick and fall apart, except we have many colors of molecules, and they have specificity in who they stick to and who they when they fall apart. And I'm just going to summarize it with sort of essentially 2 or 3 axes. The first axis is what we call competition. So imagine you have some molecules that maybe, let's say, trimerize in this case, a set of red molecules, a set of blue molecules, and each can form trimers. They can stick together. Turns out that's not so interesting. But instead, if they can trimerize with the green, and the green can participate in 2 different trimers, then the formation of this complex sort of interferes with the formation of the other complex. If all the green is sequestered away in the left, there isn't much left for the other guy to form in the first place. And if you keep taking that kind of effect further and further, and you have a complex, a supermolecule that can form many kinds of trimers, many pairs of which share some elements, you can imagine it creates a graph of sort of contingencies, where if an input signal comes and promotes the formation of that trimer, that can immediately deplete, say, the gray thing, which affects somebody else, which then promotes yet someone else, and that can cascade through in a pretty non-trivial way. And if you believe me, that's where the expressivity comes from, that kind of fighting over things. Okay? That's one axis. And so what does that require? It requires many species, and it requires that the same species participate in multiple complexes. And if you think through it, that's an interesting question. What will their interaction matrix look like? It will look nothing like the traditional biology circuits where each molecule interacts with just a couple of others. It's got to have sort of a bunch of ones in each row. Each molecule typically interacts with a large number of others, but at the same time, it can't be all ones because then really secretly there's only one species. We're still working on this, but there's some fine balance of, some kind of frustration. There's some notion of frustration encoded in this matrix that encodes this competition. Yes?
How do these look like compared to known protein-protein interaction maps?
Actually, that's a great question. So,
much more dense than
that signaling networks. So partly this is work in progress because we just know what will not work here. Certainly, low promiscuity will not work, but also just random high promiscuity is not at all the optimal thing. What you want is some kind of frustration. We're still struggling with how to quantify it, but I guess the right thing to do if we figure it out is compare to data of the type you just said. Yes. Or yeah.
Yeah, maybe I don't want to jump ahead, but do you know how this scales with number of species? Like if you have like 100, like do you expect what percentage row is consistent when you have 15, for example?
Great question. So in this old paper, we answered a different kind of related scaling question, but the precise one you're asking about this expressivity, I mean, surely it gets better, though I will say it doesn't get better. It gets better assuming you know how to find the right matrix of a scale 100. Meaning, if it's a 5 by 5 matrix and you pick a random one, it's not great, but it's not terrible. But if it's a 20 by 20 matrix and you pick a random one, that's a self-averaging property and it's useless. But if you evolve it, you can do very well with 20 compared to 5.
Like a biological, like binary search or something? Other interacting species?
No, I guess I'm just saying the larger the number of species, the better. But better in that if you have a process like evolution that can go find the rare treasures in that space. But the treasures are rarer and rarer in higher dimensional space, but they're more and more valuable when you, right? Yeah.
I think you probably said this, but so, as we climb up on the competition axis here, I'm thinking back to these phase diagrams.
Yeah.
You had.
Yes.
Essentially, we're getting more and more expressive.
Yeah. Yes.
And the axes of the phase diagrams are related to external inputs. That's how
Yes.
we're
Yes.
Well, what's the output? Like what?
Good. So in this, so the output in the context of phase diagrams are different phases that condense. So let's say it's liquid-liquid phase separation. You have a supermolecules. Sometimes some just glom together and fall out. What's in them? Sometimes it's enriched in molecule A. Sometimes it's enriched in molecule B. That's output A versus B. In the sort of lower multimerization case, it's literally which complex did you form? Did you form? This guy is output A, this guy might be output B, because they each secretly have some chemical activity that they can go do downstream. Yes?
So more networks, right? So we can prove that,
right.
Forward approximators and continuous functions.
In some large limit.
Not in some large limit. You need one layer of your operation, one relu.
If you take a large, yes.
Right. Very wide.
Right.
So what's the analogous thing here?
So I don't know. Sure. I mean, my guess would be if you make the numbers large enough, meaning theoretically take n to infinity. Well, I don't know why I think that. There's a chance that it's also a universal approximator. I know other more artificial molecular systems, people have proven it's a universal approximator. I guess our perspective was, what can you do with a realistic small number of molecules? Like, let's say you have a budget of 5. Our point is if you do it in this way, you get a lot of expressivity, but if you copy neuroscientists, you don't get much. So we're thinking more with a small finite number, what's the most bang you can get for your buck? Yes.
Is there a mapping between this and like current neural networks or like Hopfield binary weights? Or?
No, great question. I think not. It's actually going to come up in a, but yeah.
Yeah, these kind of molecular interactions are going to be restricted to the spaces within cells. And of course that
can
lifetime cell.
100%. Yes.
Many organisms have many cells, trillions even.
Well, that's
have and those cells can just states.
Okay, that's
change fundamentally the parameters.
perfect question. It'll come up in 2 slides in a second. Cell types, yes. Maybe I'll go to that then. So I talked about this one-axis competition. The next axis actually was anticipated. This was just showing literal trimerization. But imagine at any level of competition, you could say, oh, maybe they're tetramerides, maybe they hexamerides, and so on. And if you go all the way out there, essentially it's thermodynamic limit, where the things they're forming are so large, and essentially secretly that's the limit of condensates and very large assemblies. And it turns out that dimension also helps for a pretty intuitive reason that you get sharper decisions when you have higher cooperativity. And so one clear symptom of this is, in fact, this is real data from a particular cell type. This is the input-output computation, 2 ligand levels, and then what's shown is some level of some other molecule. And if you notice, though, secretly, the axes are here on a log scale, okay, which is cheating a little bit. Yeah, it's expressive, but when you change your concentrations by orders of magnitude. So this is actually a work with a collaborator led by Krishna Srinivas at Northwestern, and these are coming from phase separation models, essentially all the way out here. And you see we get just as interesting things, it's theory work, but the axes are linear scale, okay? Because it's a much more expressive system because it's many components and highly cooperative. And to somebody's question, the 2 colors here are which phase condensed. Was it enriched in a green molecule or in a red molecule? Maybe that's all I wanted to say. And because I'm running out of time, I was going to tell you, okay, so that's on this end. And to show you that this perspective has teeth, I was going to tell you briefly about essentially experiments we could do at this end, which let you do things that you wouldn't be able to do if you didn't have this perspective. In the interest of time, I'm going to be rather quick and just say the experiments involved were truly in that corner. Okay, we took a super molecules, like hundreds of species, and with a dense network of interactions. It was highly cooperative because we put in interactions that let these molecules form large assemblies through these interactions. They come together to form hundreds of molecular complexes with hundreds of molecules in them. It's also highly competitive. The 3 structures they could form, in fact, we chose them to be H, A, and M because there's a conceptual connection to Hopfield's associative memory. So we chose them to look like H, A, and M. And it's highly competitive because the same molecules are present in each of these 3 phases. These are essentially thermodynamic phases. Much like ice can crystallize in different ways, these molecules can condense in 3 different ways. And when you form one, you're depleting things from the other. Okay? And also we were in a highly kinetic regime. And the point is, with this, what you could show was you take the system, the inputs to the system are just concentrations of several molecules. Okay? And if you do the experiments with one pattern, certain patterns of concentration, certain inputs, you get one particular output, but each of the other outputs for specific inputs, much like a Hopfield associative memory, for those of you who know what it is. But what we could really show with this experiment was if you conceptualize some high-dimensional input space, which are now not really images, but concentration patterns of, say, a few hundred molecules, there are entire regions where the system will respond with H, other regions where it will respond with A, and other regions where it will respond with M. And further, we could play the expressivity game where, let's say, Ben picks 3 points, the 3 green points in input space, and picks them arbitrarily to make life difficult for me, just picks them and says, I want these 3 conditions, environmental conditions, to result in condensate H. And I want these 3, the stars, to result in a different outcome. And then we asked, can we choose interactions to put the phase boundary of the system exactly between those 2, right? And with 3 points, maybe we succeed, and then he'll challenge us with a 4th point and a 5th point until we fail, and that gives us the expressivity. I won't go into it, but essentially we did it in experiments. We also developed a theory for it, showing that these phase boundaries of a many-component system essentially are polynomial equations where the order of the polynomial is essentially the degree of supersaturation of this molecular soup, how far you are from the equilibrium, from the melting temperature, so to speak, and giving you some kind of trade-offs. But the reason I'm really showing you this is this take-home. Okay? It's a problem that many others have tried to solve, where they have some high-dimensional input, they build a molecular system to classify that in 2 phases. But Others have tried it with this abstraction first approach. Okay? And we did it by just putting phase boundaries between what we want. What do we get for that? Well, we could go to a much larger problem than others could, 900 versus something else. But then, just as important, we could use the field that does this. They use purified components. They in vitro experiments where they take great care to make sure the molecules they put into the tube are exactly what the theory told you to put in. And we did our experiments with unpurified components. And it still worked. Why? When you have a circuit diagram like that, that's like this MacBook Air. If you go change one little interaction or two or three interactions, it's going to stop working. But if you have a phase diagram of water and there are some phase boundaries and you go change a few interactions, meaning, well, at least in this multi-component system, you change a few interactions, the phase boundary doesn't move that much. Right? It's some emergent phenomena. It's much harder to move that. And so essentially, wrapping up this part, this is in some sense the promise of this kind of physics first approach rather than mimicking neurons. Nothing wrong with neurons. It's just if what you have in your tube are molecules, you shouldn't ask them to act like neurons. Yeah.
Can I ask a question about neurons? This is just making me think, okay, Eve Marder has some work where you connect neurons in certain ways, and then you can parameterize those neurons with ion channels in lots of different ways.
And there's a lot of...
There's kind of a phase space where you get certain oscillation patterns here, and maybe actually in some other disconnected region here.
Right.
It seems maybe related to this. Like, just curious.
So there would be an equivalent notion here, meaning the parameters we used are not the only ones that will give you this behavior. That's in some sense my point, that like there are many sets of parameters that will give you the same phase boundaries. You could do a similar calculation, we haven't. But, sorry, yes.
What makes you think that neurons are using algorithms anyway?
Well, maybe the more fair thing to say is...
in this, they're not?
Okay, I think that is a valid question for a neuroscientist, which is to what extent is the functioning of a brain explained entirely by the abstraction of neurons into linear threshold devices? You know, when we do an artificial neural network, a neuron is a linear threshold device. A real neuron does other things too. I think it's an open question. Does the other stuff, is it needed? Is it not? But maybe he can answer.
I was going to ask about like microbe interaction network.
So microbial?
Yeah, so if you have a bunch of bacteria that interact with each other and you can have competing and
100%.
Could you imagine building this up with like having a little like vat of microbes that are performing computations in the same way?
Yeah, I mean, we haven't thought about it for this. I'm going to talk about some kind of Pavlovian conditioning in a moment. We've tried some experiments, but so I will point out this is working not just because it's some network. It's like the physics of these molecules are important, that these form two-dimensional or three-dimensional structures. That explains this. Microbes don't form, well, they form other things. So it could still work. It'll just be it'll be exploiting the physics or the biology of microbial ecosystems. It's not going to work for a trivial reason. Yeah.
I guess I have two questions. I guess the first is a conceptual question. Is that you started off by saying that you can like one of the parameters you can change is just like the expressivity.
Yes.
I guess should I be thinking that there's sort of like either like a reward or error signal that the cell like receives?
No, that's the next part of the talk.
Okay, and then I guess like the second question relates sort of like to the interaction like network. Like, are you able, like, I guess in experiments, like track the like evolution of the interaction network?
So yes, because of a trick that I'm going to tell you in a second. Yeah.
You started off by saying that the cost, like algorithm first, the cost of computation is actually low.
Yeah, because it's a...
Yes, because you build it in an artificial way. Going back to the neuron question, right, our brain cost is pretty low compared to neural networks, like extremely low compared to neural networks, like ChatGPT and so on.
Right. But that's completely consistent with this, though, because...
Yeah, no, it's consistent. So do you think it's because the activation thing is not actually everything and we need to consider what neurons are doing, or it's basically our model of how neurons are interacting is not quite complete, or
No, I think people, at least my friends, say, no, but that's because the brain's algorithm is running and was or if you can even separate these things, is meant for the neural hardware that the brain already has. But when we run it on a computer, we're running it not on neurons, we're running it on some CMOS device. And so, for example, neural networks on a CPU are even more expensive. And neural networks, when moved to a GPU, are way better in terms of energy. And tomorrow we can get even better if our hardware is shaped. So the reason for the mismatch you're talking about is a mismatch between how hardware is currently designed and the algorithm of neural networks. And so that's what this other community I mentioned is trying to solve. Neuromorphic computing, for example, is that. Right? All right. I'll try to wrap this up. So one little aspect, I did say within a lifetime, but I kept changing all the parameters. But as you all know, you can change molecular interaction parameters, direct parameters, within a lifetime. That takes evolution. But it turns out that's a trick. In these dense networks, just changing expression levels, meaning concentrations of some molecules, is enough. And since it's a physics audience, at least putatively, I'll just explain this with, I think, a sort of simple analogy. Okay? Imagine you have a soup of 15 molecules that interact in this way, okay, with some fixed interaction matrix, Jij, JAB, that's real and fixed, cannot change without evolution. But what you're interested in, meaning what the world interacts with or the rest of the cell interacts with, are just 5 of those molecules. The input and the output molecules together form just 5. So what you often care about is the effective interaction matrix of these 5 guys, okay? Not this real one. And the key point is, even if this is fixed, this effective interaction matrix is not fixed because it depends on the concentrations of each of these hidden species. You integrate out, you have a theory with 15 fields, you integrate out 10 of them to get the effective Lagrangian for 5. Well, the values of those, the average values of those 10 fields will show up in your final result. And so this is now changeable just by changing expression levels. Okay? And here there are still open questions. The point I want to make for you is just this can be fixed, and yet this is changeable within a lifetime. There's still a question of what would evolution do if you wanted to optimize how you can change this. How should you pick this in the first place? I don't know. And this gets to Richard's question. In fact, this does happen when you compare across cell types. So the BMP pathway, it's one set of molecules, fixed interactions in all cell types, but in different cell types you have different expression levels of some of these receptors in here. And that lets you change what computation, if you wish, is done in different cell types. Okay? And actually, even in this work with Krishna Srinivas, these are phase diagrams of a condensate. I showed you a couple earlier. And these were obtained without changing any interactions, just by changing concentrations of hidden species. But you look at the phase diagram of, like, you promised me you'll only look at the phase diagram for the first 5 species. Okay? Like, that's all you look at. The others you won't ask questions about. When you marginalize over them, you get, and changing the amounts of those, you can switch between these. Okay? And so this last part I want to tell you about is essentially how can you, like what somebody just asked, how can you get a cell? This is evolution. There are different cell types. The last thing I'll quickly wrap up is, can you take this map, show the cell type examples of this input-output relationship? Okay? So meaning this current neural network is not classifying cats versus dogs. It's doing pigeons versus something else. I don't know. And then you show it examples of cats and dogs. And can some feedback process change this to that? Okay? So that's the last part. And the key point is what I will not let myself do, which is you can't use backprop. Sure, you can do that on a computer. Obviously, a cell can't do it. A cell can obviously do it by over evolutionary time, but how could it do it within a lifetime? And so here our perspective is essentially we learned from the other part of neuroscience where you change parameters not based on an obvious loss function. So backprop is like change parameters in a way that your neural network will do better, right? And evolution, at least dominantly, not if you read Richard's book, but dominantly people are like, evolution is you make a mutation if it's going to make you more fit. Oh, sorry, you keep a mutation around if it's going to make you more fit. But there's another perspective, heavy in learning. It's more like this. You have apologies to the neuroscientists in the audience, but you have a brain or some physical system. You show it some stimuli, and the system responds to it. Maybe the neurons fire. And based on those firing, based on its response, you make some changes to its parameters, okay, just locally. The most famous one is neurons that fire together, wire together, okay? And so it doesn't feel like a goal-directed process, and yet it turns out in many contexts it's somewhat goal-directed. It can move the weights in a way that improves function, okay? So that's our inspiration, because you could imagine, certainly you can imagine neurons doing this, and I'll show you what it means for molecules in a second. There's an update to Hebbian rules. So in fact, Jeff Hinton, who won the Nobel Prize last year or something, one of his contributions was developing Boltzmann machines, where he cleverly said, you can do even better than Hebbian if you alternate Hebbian learning with an anti-Hebbian phase. And let me just get this idea across to you. There's a wake phase, where there's a system, in my case it'll be a molecular system, where you show it the behavior you want and change parameters so as to reinforce that behavior, to stabilize that behavior. Okay, neurons that fire together, wire together. And then there's a sleep phase where you don't show it the behavior you want. In a supervised context, you show it inputs, but you don't tell it what the output should be. You show it a cat, you don't tell it it's a cat. And you let it dream, be spontaneous. And whatever behavior it shows, you do an anti-Hebbian update. You try to delete that from its thing. And what he argued was if you repeat this, this will converge when these two are equal. And it's a really clever idea because it's just based on response. Well, when will they be equal? It'll be equal when your dreams are exactly what you were being told to do when you were awake, which is your spontaneous behavior is what you were being trained to do. Okay? And so people have proposed this. Eric Winfrey, my collaborator on this, proposed this many years ago as a way of doing this as a way of training molecular systems. The main big obstacle is, and it's for neuroscience too, it's non-local in time. There is a sleep behavior and a wake behavior, and you've got to somehow biologically change the brain from being in a Hebbian state to an anti-Hebbian state. Andrea Liu's group has exploited this algorithm, but again, it's in an engineering context, you can get around it. They essentially have two copies of the brain, so to speak, one in sleep, one in wake. But recently, we essentially solved this problem, I think, by not needing a Hebbian and anti-Hebbian phase. The system will always be in Hebbian, but in ways I'll explain, you can essentially exploit the benefits of this. So here's a proposal for the last 7 minutes, okay? Maybe focus on the last part, which is our training rule. The equivalent of neuroscience is fire together, wire together, okay? Is going to be, we're going to assume that molecules auto-regulate, meaning if there's more of a molecule, it's going to produce more of itself, but with a high-pass filter, which is to say, if it's a static value, stop, just ignore it. Essentially, it's called adaptation, as many people at Yale know. But if a molecule's concentration is changing rapidly, make more of it. If it's dropping rapidly, make less of it. Okay? That's what this rule says. And our claim is that that one rule allows many molecular systems to do all kinds of neural-like behavior. Okay? And I think I'll have time for just one, so I'll get through Pavlovian conditioning. Everybody knows Pavlovian, right? That's a target. And I want to argue that that simple rule I said will train many molecular systems to do that. So imagine you have some soup of interacting molecules, and there's a bell molecule and a food molecule from outside, and there's one molecule on the inside that you call saliva that you will look at to know how it's responding. Okay? And here is the idea. Imagine the environment has correlated occurrences of the bell and food. Okay? The one assumption, so when the bell is high, the food is showing up. When the bell is low, there is no food. One further thing that we need is that the food shows up in this sort of asymmetric spikes. So imagine the food shows up and then slowly decays away. Shows up, decays away with two different time scales. Turns out that is sufficient because what happens is when the bell is high and there is no food, your system settles to some equilibrium state. Okay? I'm describing this in general terms because it is more general than the specific networks I'm showing you. When there is no food, the system is in some state. When the food shows up rapidly, the system is going to shift its state because they react in some way to the food. And so it shifts equilibrium fast. The key point is the training rule says, watch what's moving fast in the cell. As you go from sleep to wake, some things in the cell will change fast, some will not change. Whatever is changing fast, this auto-regulation rule will create more or less of it. And if you work through our math, it'll act like a friction term. It'll create more or less in a way that will dampen the motion of that molecule in the next cycle. Which means over time, as these spikes show up, internal concentrations will adjust so that sleep and wake actually become the same state. That is, you don't need the food to act like you're seeing the food. Am I making some sense? Okay, this is essentially the heart of the training idea. You keep jostling the cell from sleep to wake and ask what moved. And whatever was moving, make more or less of it in a way that will prevent motion in the next cycle. And almost tautologically, it means after a while you will have training. And during this process, what happens is all these other species' concentrations change. And I told you that changes function, and you get Pavlovian conditioning. Okay? You can go beyond Pavlovian conditioning. You can learn a quantitative relationship between inputs and outputs. So imagine for low amounts of bell, you want this amount of saliva, and for a high amount of bell, you want a different amount of saliva, a quantitative relationship. You can do that. Just that's your environment. You put that same cell in that environment. It turns out, this is the input, this is the output. The current response is this black curve, but instead you want it to pass through the two red points. Just expose the cell to that environment, and later the input-output properties will look like that. Making some sense? Okay. So then if you believe that, we essentially have more and more examples of that type. You can train for any input-output behavior. I'll briefly say, in biology, in microbial biology, there's this common thing called bet hedging, where a microbe can adopt one of two types, can be in one of two states, antibiotic resistant and not. And every population will put a small fraction of its population in the resistant state. And the implication is, how much should you put in the resistant state? Well, it should be related to how often you see antibiotics in the environment, because evolution would have selected communities on some larger scale to do that. Does that make some sense? And that might all be true, but it turns out the rule I just showed you, completely independent of evolution, no adaptive benefit, will train if you take a bistable circuit of the type people have looked at before, and assume one of the molecules in the circuit is autoregulatory, what you can show is, I'm going through this fast, essentially if the current distribution of the cell phenotype is the green, but you put it in an environment where it's rainy and sunny, with a different fraction, like Seattle, the Yale, I guess it's the other way in this case, but the green distribution will become the red distribution. The spontaneous distribution will shift and match how often you see rainy and sunny days, not because of any evolutionary adaptation, just a simple physical process will naturally drive it. Okay. I think I'm out of time, so I'll finish with one sort of synthetic biology motivation for why do all this. And that is that all these training ideas, this feedback, it's based on behavior. Essentially, the system doesn't behave like it should in the training phase. You keep adjusting parameters, which means it's model-free. We're not assuming any model of how these molecules actually interact. So for example, you might have thought these molecules dimerize, but maybe they trimerize or do something else in the cell. Doesn't matter. If you have this training rule and put those cells in the same environment, all the cells, those that do this and that, will get trained for the same phenotype, even though you needed different levels of the molecules to achieve that. Does that make some sense? And that's very valuable, because in synthetic biology, what people do is they come up with a circuit and then put it in the cell, and it stops working because there are some other new molecules in there that interact. Okay? And so then you have to iterate. But in this approach, that doesn't matter. You just take it and keep training it. The training is looking at behavior. Behavior accounts for all the unknown stuff. And so you will still successfully train. Yeah, and that's maybe the main point. So I guess to summarize, maybe my larger point is we know this kind of behavior happens in biology, at least on some scales. I'm not saying I don't know of a case yet on the microbial scale where we know this happens, but maybe the point is this is a proof of principle that all you need for it are these ingredients, This is a statement about the environment, some structure in the environment. This is a statement about dense interactions, trainable substrate. And then maybe most important, some kind of rate-sensitive regulation is sufficient. With that, I'll thank you and I will say this is really, I put a sort of an amalgam of work over 10 years. I don't actually know any one domain really much about anything. And so each time we want to work on something, on phase separation, we work with the experts, like a local expert from here, Mason Rouge, who was a grad student here. I used to do this on the mechanical side with a grad student who's faculty now, and Eric Winfrey, who's really been a great collaborator on much of this. So with that, I'll thank you.
Great talk. I had a question about the first part of the dichotomy between algorithmic and would it be fair to say that you're arguing for energy minimization as an algorithm to learn things rather than feed forward?
Right, so at least the examples I showed you are of that type, but I would say the larger principle is whatever your physical, like whatever the hardware you have, whatever it does naturally, often can solve those problems already. The specific molecular systems I looked at were just energy minimizing systems. Even that's not quite true because the experiments I showed you, this pattern recognition thing, it turns out it's in a kinetic regime, so it's really not minimizing an energy, it's instead solving a min-max problem over trajectory space, but whatever. Right? Yeah, but roughly I agree with you. Each physical system has its own thing it does naturally, and you can exploit that. And you pay, you gain in sort of complex, or you gain in sort of compactness, I should say. Yeah.
Is there a simple intuition for why heavy learning alone wasn't enough in your case?
Yeah.
You need this contrastive.
Contrastive. The intuition, so the citation disappeared, but all of you know Jan LeCun. Jan LeCun has this interesting set of YouTube lectures, a 3-hour lecture called Energy-Based Learning or something, and it's essentially all about this. The intuition is, Hebbian is just a positive reinforcement, so it can teach you, hey, a cat, I should respond cat. But there's no negative reinforcement, so if by mistake you associate during the training phase a cat with a dog, there's no phase that ever moves weights in the opposite way. And so, I don't know if I even have a slide, so maybe I don't need to show this. But the rough intuition is if all you did was Hebbian, you will get false positives in some sense. And the negative phase could be anything. I showed you one thing. Our earlier papers on mechanical systems, we did something even simpler. Like something as simple as do Hebbian learning, but while you're asleep, just scramble some fraction of your weights. Okay? Like Hinton's idea is something more specific. Like you have dreams when you're sleeping, and based on those dreams, forget stuff. No, it can even be random forgetting. Essentially, you're forcing the system to relearn stuff, which means the stuff that's persistent, you're going to truly learn, and the stuff that's fluctuating, you're probably not going to end up remembering.
Can I ask you, this particular slide here, was the shape of the green food spike critical?
So yes and no, yes, because this, I needed to go up fast and come down slow because essentially all the learning is happening in the fast sweep up. Essentially, you're comparing what's changing in my system, what's different about what I want to do and what I'm being forced to do, and I'm taking the difference and updating. The problem is if the downward thing was as fast, then the same learning rule will cancel it on its way back. So you'll end up not updating. Essentially, we're using the time domain to move secularly in one direction. If that makes sense. So if it's truly symmetric, it won't work. Yeah. So yes and no. So this is what, what Hinton showed was when you do this, you end up minimizing the Kullback-Leibler between the distribution of states that the system naturally wants to be in and the distribution you're showing it in the wake phase. And so he showed that that's true in some mathematical sense for neural networks. We just copied that math. So for neural networks, that rule is based on fire together, wire together. But it really comes from a mathematical principle. When you copy that math principle to molecules, the rule becomes auto-regulation. And so when you put all that together, yes, auto-regulation will minimize the Kullback-Leibler between what the system wants to do and
I'll ask one last question. So how seriously do you take the term sleep-wake cycle? Do you think this is what's happening during sleep?
So my understanding is that's why he termed it that way. And my understanding is also that people don't pay attention to that. Like people like him don't pay attention to that anymore. To me, but okay, on a more serious note, like the difficult thing about it is it's saying that the chemistry of the brain is completely different in your sleep, because like there's a signal called coincidences in firing rates. During wake, apparently you take that signal and you reinforce interactions. During sleep, you have to do the exact opposite. For a theorist, the piece of paper, sure, it's a flip of a sign, but like at the molecular level, you're asking for the opposite process. Like, it seems difficult. I think that's what we kind of solved with this thing, but my sense is people don't take it seriously.
Well, if there are no last questions,